Today I'm going to talk about work we've been doing for a long time now on how we can model the functional architecture of the human brain based on functional MRI data. I'm going to try to step you through this, the logic of this project. And I'll skip over this. So first I'm going to talk about the human brain and then using naturalistic cognition for experimental investigations. And I'll be arguing that information is encoded in fine scale patterns of neural activity, fine scale at the spatial resolution of fMRI. So that's a few millimeters. And there's a lot of information that can be gleaned from these fine scale patterns. But the patterns are idiosyncratic, so each brain has a different pattern for the same kind of information. So we needed some method for aligning the information across brains so that we can study that. And we developed a method that we call hyperalignment. And I'll conclude by saying that the transformation matrix that we derive from hyperalignment can be thought of as a digital key that unlocks an individual's neural code. So first, the human brain is arguably the most complex object in the known universe. So all of our experience, memory, knowledge, language, emotion is a result of brain activity. It is the embedding space for our mental lives and cognition. In more modern terms, you can think of it as a data center for natural intelligence. And like data centers for artificial intelligence, the brain is an energy hog. It takes a ton of energy. So it's, the brain weighs about 1,300 grams, or about 2% of body weight, but it consumes 20% of the body's energy. And it requires this energy because neural activity involves the potential energy in the resting state neuron membrane potential. Every time there's neural activity, the membrane potential is depolarized, and repolarizing the membrane potential requires a lot of energy. So now rapid increases in local blood flow provide the substrate for glucose metabolism. And this local blood flow control has a relatively fine spatial resolution. I'm talking in my world of fMRI. Less than a millimeter is fine scale resolution. When we talk about single unit recording, it's not so impressive. So one voxel in an fMRI scan probably contains hundreds of thousands of neurons. But nonetheless, information, we're aggregating information from the neural activity from 100,000 neurons, still gives us a lot of information. We can measure these local blood flow changes with functional magnetic resonance imaging, fMRI, as opposed to controlled cognitive experiments in the research I'm going to describe. We study how the brain deals with the world as experienced. The world, as we experience it, is complex and dynamic. It doesn't come to us divided into experimental and control variables. But the brain takes advantage of these codependencies between different events in the world and deals with them very naturally and effortlessly. And a lot of the information comes from these codependencies. And because with more modern big data methods, it's now possible to disentangle information that's encoded neurally during naturalistic cognition. So we have people do things like watch a movie or listen to a story in a relatively free-form way. And then we collect our data and we're able to make sense of it remarkably. Let's see if I can get this to work. So this is an example of one of our stimuli. We have people watch the Grand Budapest Hotel. I hope you've all seen it. It's a wonderful movie. This, on the left, shows two brains, the patterns of activity that we measure with fMRI in two brains while watching the movie. And they're in sync. So these two people are watching the same movie, having we assume similar experiences, but the patterns of activity look quite different. I'm stuck. So the data from fMRI are put into a two-dimensional matrix, and in this two-dimensional matrix, each column is a cortical vertex, and each row is a time point. So this is the opposite of the way you do your data. So we can analyze these data in terms of the time during activity at one point in the cortex, which is a response profile, or we can look at a pattern of activity across vertices at any point in time. And the data that I'm going to show you use these response patterns for neural decoding. So we think about each the pattern of activity at each time point as a vector, and that vector can be analyzed in a high-dimensional information space where each vertex, cortical vertex, is a dimension. And then each time point in the movie, it has a vector in a different location in this high-dimensional information space. And we can then use the data in this space for decoding using machine learning. So this is a study where we had people look at 6 different species of animals, 2 species of insect, 2 species of bird, 2 species of primate, and we used a machine learning pattern classifier to support a vector machine to predict which animal the subject was looking at based on the pattern of activity that we measure with fMRI. So this is a confusion matrix. So the viewed stimulus is along the x-axis, and the classifier output is along the y-axis. And the diagonal shows correct classifications. So the participant was looking at a ladybug, and the classifier said it was a ladybug pattern. Or the participant was looking at a yellow warbler, and the classifier said it was a yellow warbler. So the overall accuracy is about 70% because it's a 1 out of 6 classification. The problem is that we build these classifiers based on within subject, within brain analysis. So we built the decision surface for the classifier based on training data, and then we test the validity or the accuracy of that classifier on data in the same subject. And we tried to do this across subjects. So we built a classifier based on, say, 20 subjects, and then try to classify the patterns of activity in the 21st subject. It doesn't work because these patterns of activity don't correspond across subjects. So the level of accuracy drops from about 68% down to 37%, and the confusion matrix looks terrible. And if you look at this across the whole brain, you can see that within subject classification is very high in occipital temporal cortices, which are the visual areas, and the between subject classification drops drastically and is only significant in very early visual areas. So we developed this method called hyperline to try to align the information across brains. And the idea is that if we have 2 brains who are experiencing the same stimuli, same naturalistic stimuli, in their information spaces based on the cortical vertices, the locations of the vectors for each time point are out of alignment. So hyperline finds a transformation matrix that transforms these individual information spaces into this common model space. So this is the embedding space for cognition. So this involves 2 things. First of all, we have to derive the common model space. And this is computationally intensive. I just did an analysis with deriving common space for 20 brains, and it took about 2 days worth of CPU time. And then once we have the common space, we've derived these transformation matrices, and that's also computationally intensive, taking about an hour for each brain. So it looks like this. We can take we have a brain data, a data matrix, and we derived this transformation matrix R, and we simply multiply these to project the brain data into the common model space. And the goal here is to find transformation matrices when multiplied by brain matrices minimizes the difference between the projected data and the mean data for the group. And there are a variety of ways to calculate these transformation matrices. We've been using Procruste's transformations for years, and they work very well. And more recently, we found that we can use ridge regression to get even better results. But they're linear methods. So this, in the domain that I'm talking about, linear methods are extremely effective, and we're very scared to go nonlinear. So if you think about this scheme, we can take the brain data and we can decompose it into the model and the transpose of the transformation matrix. So this is really saying that the individual brain data can be decomposed into the shared information in the common model space and the transformation matrices, which capture something about the idiosyncratic topographies that carry this information. So the shared information, this just shows that we can derive the transformation matrices for brains based on their movie viewing data, then apply those transformation matrices to the experimental data for that animal perception experiment that I showed you earlier. After we do that, between subject classification now looks almost identical to within subject classification. And in fact, it's a little bit better because we have a larger data set for the training data from multiple subjects rather than a single subject. And the transformation matrices capture something about the idiosyncratic topographies. So you can take data from the common model space, so a vector from the common model space, and multiply it by the transformation matrix, and have that vector transformed into the vertex coordinates of the individual brain. So for example, we could take the vector for the average response to faces and from other brains and put it into a new brain's cortical topography. And we can similarly, we can take the vector for the response to objects and project that into this one brain's idiosyncratic topography. So this is what idiosyncratic topographies look like. This is the face selective topography in ventral temporal cortex in 3 different brains. And like people have shown for decades, there are face selective patches in the fusiform gyrus in the ventral temporal lobe. But the precise location and shape of these areas differs by subject. If we take other people's functional localizer data, the location of their topographies, and then use hyperalignment to project those topographies into each of these individuals' brains, we can then estimate this topography. Now, this is a topography estimated based on other people's face versus object selectivity. And you can see it looks just like the face versus object selectivity based on that subject's own data. So that transformation matrix has captured this individual variation in functional topography. And then finally, once we've found the common information using hyperalignment, then the variation around what's shared across brains it comes into focus, the variation in the fine scale pattern. And so we can do things like predict intelligence based on patterns of brain connectivity in the fine scale after hyperalignment. And if we use the hyperaligned data we can account for twice as much variance in intelligence than if we do it based on anatomical data. So this method also allows us to look at individual differences as well as the shared information across brains. So the take-home message here is that this transformation metrics seems to have something kind of magical about it. And we can think about it as a digital key that unlocks individuals' neural code if we have data in a common model space from other people's brains. So we can decode the neural activity patterns in an individual based on the activity patterns in other people's brains after they've been hyperaligned. We can estimate the individual specific functional topographies in this brain based on other subjects' individual topographies. And we can model individual differences in things like cognitive ability or other psychological traits. And that's what I wanted to say. So thank you very much for your attention.
Thank you, Jim, for this amazing talk. We have time for a couple of questions. Yes?
So I'm curious, you talk about all these idiosyncrasies between brains. Are twins more similar in their brain idiosyncraticness? And then if they are, can you use that to sort of fine-tune the method that you've developed?
We've done an analysis of twin data, and we find that the fine-scale patterns are not very heritable.
Oh, what about
So the core scale patterns are heritable.
Okay.
And so we really think there's a fundamental distinction between the core scale information, the core scale organization of the brain, which is heritable, and supports things like the location of brain areas.
And
where
is that also, given the environment being similar, in which the brains are raised versus not raised together?
We haven't done raised together, or we haven't looked at that. But we've done, compared monozygotic to dizygotic twins, sort of classic
Yes.
behavioral genetics.
Right.
And
Because if it was actually segregated versus were they raised together in similar?
they were raised together.
Yeah, okay.
But this is from
Yes.
there's a public database called
so
ABCD.
So it's not very heritable.
It's not heritable,
Yeah.
but as I showed, the fine scale information carries information about much finer distinctions in information, about information processing, and intelligence. So we think that the fine scale information develops based on experience and learning more.
Great, that's awesome.
We have two more questions.
Nice. This is very interesting, but is this going to render torture useless? In other words, can you find the key to the brain of the individual out of your transformation matrix?
Pardon me?
Well, let me repeat. Is this research ultimately going to render torture useless in allowing you to have the key to an individual's map of reality?
Well, that's a total evil thought.
You know, now with artificial intelligence, there's all sorts of evil thoughts.
Yeah.
I'm coming from a totally different area, but I find this very impressive.
Now, there's potential for reading people's thoughts, for detecting lies, things like that. And all of those potentials are unrealized, but I'm sure some evil lab somewhere is going to try to do it.
I was curious as when you find a matrix that is common to all the subjects, how many dimensions do you actually need to keep to do a good job?
For a brain area,
Yeah,
so
depends on the brain area.
A few square centimeters of brain, we need about 30 to 40 dimensions.
That's for your visual data that you showed us.
For these visual data. For the whole brain, we can do this. We can predict which time point in the movie someone is looking at based on the similarity of the pattern in that subject's brain while they're looking at that moment in time in the movie to everybody else. And the overall accuracy is about 95%, which is kind of frightening.
Amazing. With 30, 40 dimensions.
No, but that's for the whole brain, it's about 400 to 500 dimensions.
Still very small.
It's still very small, yeah.
Yeah.
But we're working with fMRI, so, it's.
Amazing.
Yeah.
Thank you.
Okay. Yes.
So when we work, I work with the brain-machine interfaces, but usually with intracortical recordings. And so we are used to have subject-specific decoders, okay? But I don't think it's big, as we were discussing before with Professor Solla, I don't think it's because they are inherently different, or even if they are, it doesn't really matter, but because having intracortical recordings, you have a very small window into what's going on. So we have different, it's more differences in the recording than in the actual structure. I was wondering, in fMRI, so you think that you also have an element that, they're not perfectly aligned, or the heterogeneity is completely due to heterogeneity in the actual object, or you also have heterogeneity in the way you measure the object?
Well, there's heterogeneity in the topography.
Okay.
So the patterns are different, but the same information is in that topography. Now we have another lab at Dartmouth that has done hyperalignment on single unit recording data in the hippocampus and shown that it's effective there. So it's conceivable that it would be possible to build a transformation matrix for individuals neural population for this small sample to be able to get with a brain-computer interface and find what's common and then expand the power by using data from other monkeys to decode that.
Thanks.
Nice.
Okay.
One more.
Yeah, sure.
Have you considered people with neurodivergent brains compared to neurotypical brains?
We haven't done that yet, but yes, we certainly are thinking about that and trying to come up with a better model for what is the transformation from neurotypical to neuroatypical. We're currently gathering data in monkeys while they watch a movie. And we have humans watching the same movies. We're going to try to do a transformation from human to monkey. We have nothing to report on that.
Sorry, what?
That's very neuroatypical.
What was the movie they were watching?
We used a Disney movie called Monkey Kingdom. So it's about a troop of macaques in Sri Lanka. So we don't know if the monkeys liked it or not. That was our best shot.
Not yet.